The overall goal of this procedure is to determine the structure of macromolecular complexes by single particle analysis of electron cryo-microscopy data using the image processing software SPHIRE.
Single particle cryo-EM is becoming a mainstream technology for studying macromolecule structures at near atomic resolution. SPHIRE is a novel software package that enables semi-automated processing of cryo-EM data in a user-friendly manner.
The main advantage of SPHIRE is the automatic validation mechanism. Most of the procedures need to be run only once, and the results are objective and reproducible.
High-resolution cryo-EM can be used to place individual residues, thereby providing crucial insight into the organization and function of complex macromolecular machines.
Individuals new to this method should keep in mind that obtaining a high-resolution structure highly depends on the quality of the sample and the input data.
To begin using the SPHIRE application, open the GUI from the terminal. Open the utilities to display a cryomicroscopy image with E2 display. Use the built-in measurement tool to measure the longest particle axis in angstrom per pixel. Open the project settings. Set the CTF window size to at least the particle box size. Set the particle box size to an even number at least 1.5 times the particle diameter. Set the particle radius to at least half of the measured length. Enter the point group symmetry if known